Good afternoon, everyone. On behalf of Ana Vázquez Parrado, Director of this Center, and on my own behalf, I would like to deliver you a short welcoming speech to once again congratulate on the opportunity of resuming our program, our cultural program, this time on site, as we have long expected. So, nearly after a year after having had the pleasure of hosting the event Deciphering of the Human Essence, that gathered together paleontologist Juan Luis Arcebuaga and neuroscientist Axel Clearmans, we are given the opportunity to welcome you on a very special occasion. As to today's event, we are happy to present the talk Technology in Medicine, What Does the Future Hold for Us? As an event within the series Spanish Influencers in Science that CEBE organizes for the 5th time in collaboration with Instituto Cervantes and, of course, with the Cultural Council of the Spanish Embassy in Belgium. So, as you all know well now, all three of us, all these three institutions, together with the Office of the Higher Council for Scientific Research, join forces to develop and coordinate a program under the Spanish Scientific Diplomacy Framework, which is locally labeled in Belgium as Spread Science, that beautiful program that has been conceived of by our embassy, and we all follow up. So, very briefly now, this event will address the impact of technology on the future of medicine. To enlighten us on the topic, we have the pleasure to count on Professor Lechuga Gómez. Muchas gracias, Laura. Thank you. Researcher at the Higher Council of Scientific Research and National Research Prize for 2020, thanks to her work around la on-chip. And on the other hand, Professor Liesbet Geris, who is a remarkable researcher at the University of Liège and Leuven, at the KU Leuven. Is a specialist in biomechanics and materials engineering. So, let me now finalize my welcome words by thanking our guests, once again, Laura, Lechuga and Liesbet Geris, for your interest and presence tonight, thanks to CEBE, and very particularly so to Ana, who is leading the group now, and will take will take the floor in a minute. For your continued and successful effort in making your series forever interesting. So it's a pleasure to be once again collaborating with you. And of course, thanks to our cultural counselor, Sergi Farrell, for the support of these events within the science diplomacy framework and in the already mentioned label Spread Science. And finally, our sincere thanks to all of you who having so cleverly chosen to be with us in this most auspicious occasion. So that goes for those online as well. So with nothing more ado, please, Ana, the floor is yours.
Thanks a lot. Good evening, everybody. Buenas tardes. Gonzalo has made my work very easy because I wanted to introduce Laura Lechuga, not Gómez, I'm sorry, Laura Lechuga and Liesbet tonight, but Gonzalo already did. So I will just say that a couple of words about our association. So I'm president of CEBE. CEBE is the Association of Spanish Scientists in Belgium. And like us, we are a lot of associations. At this moment, we are 18 associations around the world of Spanish scientists abroad. And one of our aims is to bring science closer to society and also to make a bridge between Spain and abroad, so in this case, Belgium, in scientific matters. So that's why we are here tonight. To celebrate this event about science communication, about this topic, medicine and technology. We're here tonight after almost after the end of a pandemic. And so technology is in the backbone of medicine today, and tonight we have the pleasure to have here Liesbet, who will talk about tissue engineering, and Laura, who will talk about biosensors. And so with that, I give the floor to Liesbet. Just some words for the people online. We don't forget you. So if you want to ask questions, you can use the section of questions and answers. And please put your questions there. We will, you will have your voice tonight too. Yes. Right here. Yeah.
So good evening, everyone. It is a very big honor to be invited here tonight to speak in front of you and to speak about something that one is passionate about. That's always a pleasure. So I really hope that I can do justice to the theme you've given me. And since CEBE is also interested in bringing science closer to the public, I thought I'd start with a little story. But before I talk about any of the work that we do or that other people do, I feel I need to show these people because they're the ones that are actually doing the work. There's also one Spanish scientist among them in my team, so lucky me. And the story that I'd like to start with is one from Rose. Rose is a small child, 8 years old. And when she was born, she had one curvy leg. Nothing special, babies always have a bit of curvy legs. But in her case, the one curvy leg didn't straighten while she was growing. And it bent even more and more, and at some point, it broke. It just snapped. She didn't fall, there was nothing going on, it just broke. And it seemed, and it didn't heal afterwards. Not with the best care that she could get from any of the surgeons here in Belgium, the bone did not heal. And as it turns out, she's one of the 500 children in Europe that has a genetic defect that causes a piece of bad bone at that location in her leg. And so the only solution that we could do was wait until she was old enough to get very invasive treatment with a ring fixator and bone transport, all very invasive, not something that you want to give to a child, but that is really the only solution. Unfortunately, in her case, that was not enough. So after 8 years of walking around with a broken leg, they decided to go for an amputation. So this is not something that you'd expect from a broken bone to go to an amputation. What she would have needed probably was a piece of healthy living tissue that could have been implanted instead of that bad piece of bone that she had there. And like her, there are a lot of people waiting for some tissue or some organ to replace their own missing or malfunctioning organs and tissues. And as you can see on the slide behind me, there is a very big discrepancy and a growing discrepancy between the amount of people that are in need of new tissues and new organs and the amount of donor material that we have at our availability. So part of this solution is trying to create living tissues, living implants in the laboratory. And that is exactly what tissue engineering is about, trying to create living implants that will integrate completely in the body of the patient, so that also for growing children, there are no problems that you otherwise would have with implants of metal or plastic that do not grow as the child is growing. So this is what we want to do with tissue engineering. Now, how should we do that? Well, in tissue engineering, if you try to look for examples, and we're very lucky because Mother Nature is a tissue engineer par excellence, because she has spent millions of years optimizing the way she creates tissues and she creates organs. So we can look at her for inspiration in order to try and understand what we should make in the lab that would, once we implant it, actually lead to a functional tissue. And there are two main lines that we can follow. And one is trying to recreate bone as the end product. So we look at bone, we try to understand what does it look like, how is it built, what are the materials, and try to recreate that and implant it into the body. Now this works to a certain extent and in certain situations, but there is some variability. And if you really need larger volumes, this approach is not really the best one to follow. The other approach that you can then follow is to try, rather than recreate the end product, is to try to mimic the process by which Mother Nature actually makes the tissue. So in development, when your bone is being formed, it starts with a bunch of cells. And these cells then come together and form what we call a template, something that looks like a little mini bone, but it is made of cartilage. And this cartilage then matures, a little bit what you also have when you break your bone. It's a bit the same process that takes place. It matures and it grows, and in the end it forms your bone. And what is left of this cartilage is the growth plate, which is the piece of tissue that makes sure that you can grow from a toddler to an adult size. So we could try and recreate this growth plate in the laboratory so that when we implant this will kind of be an engine for bone formation later on. So these are our two options. Now, how do we address that? There are a number of basic building blocks that we can use, that we can play with, like Lego bricks, that we can pick up in the lab and that we can combine. And I've listed 4 important elements here. And before I go into the technologies, I'll just briefly introduce these important parts. Starting with the cells. We work with stem cells, so cells that can transform themselves in any sort of tissue-forming cell. Now, these cells, for bone, we're quite lucky because we have a number of sources that we can use from the patient that are easily accessible. But if you want to have a stem cell from the heart, that is not so easy to access. So then there are maybe other options like induced pluripotent cells. Those are cells that, for instance, from a skin cell, you can push a little bit back in the developmental cascade so that they start behaving again like a stem cell that can produce multiple other types of cells. So there's also some technology involved, but I'll not go into detail there. Now, if we want to push these cells into doing something, anything, if we want to push them into becoming a cartilage-producing cell or a bone-producing cell, we need to stimulate them somehow. And the way we stimulate them is with growth factors. Growth factors are chemical substances that really will induce the cell into a specific trajectory. Now, the problem with that is, well, we have a number of growth factors that are approved for use in patient, not so many as we have that we can use in the lab, but there are a number of these. The only issue is that it's very hard to be as precise as Mother Nature is. So often you'll find that people are getting larger doses of growth factors, so you could have side effects. And also, of course, these are not free of cost, so there are some considerations there. Now, when you put the cells and the growth factors together, the way to deliver this in the body is often, especially with humans, when we have larger volumes, is by using some kind of carrier structure, something that we call a scaffold in tissue engineering. And the scaffold can be made out of various materials, and I'll show a few examples later on. But these scaffolds can also participate in the process. So it can just be an inert carrier structure, but it could also be activated. So if you give it specific properties, it can be an active contributor. It can be degradable. Ideally, it should be degradable if you want that ultimately the only thing that is left in the patient is his or her own tissue. So then it should be degradable. But it should also have the right mechanical properties in order to sustain the tissue growth. And it should be manufacturable. So we should be able to make it. And I'll come back to that later as well. Now, this whole thing then is implanted into the patient. And there are also a number of things to take into account from the patient side. Because these patients, we need to make sure that there's a right mechanical stability, that they don't squash or crush the implant when we put it there, that there is sufficient blood supply. Otherwise, anything that we will put in there will die immediately. We should also take care of the fact that these patients might not be the healthy patient. If they were the healthy patient, then probably they might not need a tissue engineered solution. So they might have diseases or they might have genetic defects. Those things we also need to take into account when we design our tissue engineering construct. And then finally, the immune response, the immune system. If the implant is made by the cells of the patient, the immune response will not be so bad, but you're still implanting maybe biomaterials that will evoke a response. And we learn, the more we do this, the more we learn about how important the immune system is in the whole process. Now, in order to bring all of this together, we need enabling technologies. We need technologies to realize these things, to work on individual components, but mostly to bring everything together and to bring it to the patient. And there are 3 types of technologies that I would like to address today that are especially used in tissue engineering. The first one is 3D printing or additive manufacturing to create the scaffolds, to create the carrier structure. The second one is about manufacturing. How do you make it? How do you create enough cells to go to the patient? Because for a mouse, we might need 1 million cells, but for a human being, we might need 200 million cells or even 1 billion cells, depending on the size of the construct. So this is a huge amount, and we need to be able to make this in a reliable way. And then finally, something that is only now gaining traction, but that is my particular passion, is the use of computer modeling and simulation to really help with all the different elements along the process to be more rational and to make better choices rather than to do trial and error. So starting with the additive manufacturing, this is a very, very simple schematic of what additive manufacturing does. Basically, it adds layer by layer until you have your final structure. So you start by making a design in the computer, you put that into a specific source code, and then what happens is that the head of this printer, if it is, for instance, an extrusion printer, it looks like a syringe where hydrogel comes out, and you can just, basically draw the structure that you want to have layer by layer and create a three-dimensional structure. You can have also other types of additive manufacturing where you, for instance, have layers of powder, metal powder, other types of materials, and then a laser will draw the picture and that will melt the powder, and so you build up a structure. So this is, the concept is layer by layer, and that's why it's called additive manufacturing. Now this is not really new. There has been a lot of work, especially also in Belgium, with a number of spin-offs of the KU Leuven that have gotten really big in this additive manufacturing. And one way this is often used already is to plan surgeries. Here you see the model, a printed model of a young patient's heart, and the surgeon uses this to be able to feel the heart, to look at it from all sides before the surgery and to better plan the surgery. Another way 3D printing or additive manufacturing is often used is in personalized implants. This is a hip implant, the part that goes into the pelvis, where your hip bone articulates. And for some patients, when they have big pieces of bone missing or more than normal bone missing, they can't use the standard implants, and you need to 3D print them based on medical images. So this is already in place. Now you can do other types of technologies, as I said, with the hydrogel, so the soft biomaterials that you can use, and you can really make different structures, different activations. You can have different combinations of cells at different places. So what is being printed can either be cells alone, cells with a hydrogel, or hydrogels alone. And you can really decide what should go where. Now, the biggest challenge at this point in time is still understanding what should go where. Having the right blueprint to print, Mother Nature hasn't been so forthcoming in giving us all the details that we need in order to create these tissues. That is how we can make certain structures. Now, if we want to include the cells, as I said, there's a huge amount of cells that we need. There is also the way that we culture the cells in the lab. We take them out of the body, we put them in 21% oxygen. This is an environment that a cell has never seen or felt, so they're a bit unused to that. So we need to understand what it is that we do to the cell will influence the cells and how we can create the best environment. And I'm just briefly going to show a number of different environments that we can use to have a better understanding. Starting with something that Laura will explain in much more detail, I guess, something that is an organoid chip. This is one of these channels that you see is 1 centimeter. So it's really on a tiny scale to try and understand certain mechanisms of how different cells interact with each other or with electrical or mechanical stimuli. We can also go a bit bigger, and now we're talking about the size of maybe 10 centimeter in the width of the box that you see, which is a tabletop bioreactor that we use to really understand how fluid flow and how certain culture regimes are affecting our cells that we culture, how the scaffold on which we put them is affecting our cells. So this is used to better understand, and in order to then use this understanding to go to the big brothers of these tiny bioreactors, and these are commercial bioreactors where we're talking about sizes of 1 meter and several hundreds of millions of cells that can be cultured in these environments. And these are already commercially available, so this is already being used, but sometimes with insufficient understanding still of exactly what is going on. And can go even one step further, and that would be to these several meters these whole chamber robotic systems, where it is no longer our lab technicians that need to work with these machines, but that the robot is doing the culture in a fully GMP room, so it's a clean room. Everything is according to proper standards, so that what comes out can go to the patient with the least amount of variation and other safety issues. So this was for technologies in terms of manufacturing. Then we come to the computer modeling, and I'm going to show you a few examples of the work that we do in the group that links to the two other examples that I showed of the two other technologies. I just wanted to mention that what we use when we say computer modeling and simulation, we sometimes also call it in silico modeling, because you have in vitro in the lab, in vivo in a living tissue, and in silico is in the computer, referring to silicon, the main component of computer chips. Now there, in silico modeling, this will also maybe ring a bell, or some of the other words might ring a bigger bell, like artificial intelligence, that is now sprinkled around as the magic solution to everything, including in healthcare. Well, it is one of the technologies that can be used on the in silico spectrum. It just depends on what information you have. When you have a biological question, you need to investigate what is the question that is being asked, what is the data that I already have, what is the information that I have, what is the knowledge that I have, and based on that you need to decide what is the right in silico technology to use. Should I start only from the data side, and then you can use artificial intelligence, machine learning, where you don't make a lot of assumptions on the process, on the biological process, or maybe I don't have enough high quality data, but I know something about the mechanism. I know how my cells interact with the environment, or I know something about how my hydrogel affects my cell behavior. So then I can use that information and translate that into mathematical equations and make a computer model that way, so that we call a knowledge-driven model. So again, it is really the availability of data and the question that needs to be answered that should determine what kind of technology to use. And just a few examples. I talked about how it is important to influence cells and steer them in a certain direction. Now we can use growth factors, I mentioned that already, but what growth factors should we use? And how should we really steer the cells? What is the best path to use? If you look at cells, how they determine their cell fate, there is a whole network of interaction between different genes, between different proteins that is active. And it's far too complex to try and understand that, work that out in your head. So we're developing these kind of network models where you try to map, really trying to create a Google Maps for your cell, and you map that out, and then you try to find, okay, my cell is here, and I want it to go there, so what would be the best way for the cell to get there? And everything that you do in silico, and here's just a small example of the same experiment that we did in the computer and in the lab, you can see that the overall trends are the same, you just have to look at the colors. But the problem is that here, this takes really weeks and weeks and weeks to create these few data points, whereas this was done in half a day. So there is it's much easier to do something in the computer and then test the best solutions in the lab, and rather than doing a trial and error approach. Now, using a computer model, we also recently designed, or we suggested to our partners that are interested in dentistry, in having implants, that the best shape to use for their new biomaterial would be something called a gyroid structure. So it has a very specific shape that you can see here. This is 3D printed in a material that looks a lot like the material that we have in bone, so calcium and phosphate. And the choice of this structure and exactly the right dimensions of the pore sizes and the thickness of the walls was determined by a computer model. You can see some steps. The green here is the tissue that is formed according to the computer model. And this is now at the stage where we're finalizing the large animal experiments and probably are going to the patient with this new scaffold structure. Now, another thing is, I talked about bioprinting. When we bioprint, we take our cells and our hydrogel and we push it through a very small needle, and then the cells, well, we hope that the cells come out alive. That's basically what a lot of people test at the end. Ooh, are my cells still alive? Then it was successful. Well, maybe we can beforehand try to understand what kind of settings we should give to our bioprinter in order for the cells to survive. And so here, this was a video where we simulate the cells and look at how long they stay in our needle, in our print head, and what kind of mechanical pressure, shear stresses they experience. and then we can work out how well they would survive the process and how we should adapt the process in order for more cells to survive. And then finally, I talked about the bioreactor. The bioreactor, which is a closed environment, so it's really hard to go and open it up and then look at the cells, how they are behaving. This is a digital twin, which is kind of the similar process that happens inside the bioreactor, but now modeled using a computer model. So here we have the schematic of the bioreactor, and it is this bit where the flow is passing through the 3D scaffold that we're here simulating. And the pink is really the cells and the matrix that they are producing, influenced by the local curvature, by the oxygen, the glucose, the shear stress of the flow. So all of these elements that we have found in the lab to be important, put into the model to simulate the process. And we've shown that this really recapitulates what we see in experiments. So this can help us to better understand what is going on. So this is a very brief overview. And I'd like to end, and I have 20 seconds left, so I'll go 1 minute over time. I'd like to end with 3 challenges that I think, regardless of what type of technology we're working on in medicine, are really important to go forward. And the first one is language and quality. It's really easy to communicate to people who exactly understand what you know in your field, and to be maybe less precise because people will kind of know what you're talking about. But when you want to go and take something to the patient or take something to industry, you need to pass certain thresholds like regulatory approval, which means that you need to communicate to people who are not used to communicating to you. So it's really important for us scientists to learn how to be precise, to avoid this Babylon Tower situation, and to make sure that when we communicate very precisely, we can also be very precise about the quality and the demands, the expectations of the regulator and what we have done. That is important. A second challenge is that for a lot of new technologies, the known rules do not apply. The way for regulators to approve this, the European Medicine Agency, if they're confronted with a computer model, they're not used to seeing that. So they don't really know how to evaluate the credibility, how to make sure that what you are showing there in your model is actually true. So we need to work with the regulators as academics, as industry, to make sure that they understand what it is that we do and how we make sure that what we are showing them is actually to be believed. And that means that we need to work with them to establish these guidelines. And we've been doing that for the past year for the Anticoagulant part. We've also been working with FDA on that. But it also means that we need to establish, if not a common language, that goes back to the first challenge, at least we need to have an understanding of who means what by what term, so that we can adapt the way we communicate and make sure that, that there is no mistakes happening there. And then finally, complementarity versus competition. I already mentioned you have the AI can solve everything. That is one of these competitive elements in silico, but also in tissue engineering, depending on where the people are coming from, you'll see that they propose biomaterials only solution or a cell-based solution only, simply because that is the world that they're coming from. Well, it shouldn't be about that. It's really the question. It's the application that should determine what kind of technology is the most appropriate to use. And of course, there is always going to be variations that is possible, but we should do away with this competitive element because it is about helping the patient. It is about making sure that we're not trying to push the technology into a living implant and then onto the patient, but that we look at the patient first and that we from that derive what is the need of that patient and what is the technology that we need to provide that solution. And only then we can create a solution for Rose and we can make sure that the next girl like her does not have to amputate, but that can use a living implant like this. Thank you very much.
Well, good afternoon. First place, I really like to thank the invitation and the opportunity to be here because I have been visiting other associations with Spanish scientists abroad, so this is a pleasure for me to be here today, also in Belgium. So what I am going to tell you is about one specific aspect, in my opinion, of the future of the medicine, that is the clinical diagnostic. So how we are going to handle the diagnostic, especially after the post-pandemic time. I'm saying that because probably what we have seen during the pandemic, probably this is very familiar to you. probably during the peak of the pandemic, we saw very long lines of people trying to get a PCR analysis. The main problem is this PCR is a very, very useful technique, a very good technique, but the problem is mainly centralized. It's mainly centralized in the laboratory. And that's the reason why we get this long line, but also long times to get a PCR. Even a PCR you can run in 2-3 hours, but even during the peak of the pandemic, if you remember, takes even, for some people, several days, even 1 week to get a result. So this is the problem because the clinical diagnosis is mainly centralized. And it's also happening not only for the PCR, as we saw in the pandemic, but it's for any kind of disease. For example, if you don't feel well, you go to see your family doctor, and normally he's going to ask you, okay, we need to go some blood analysis, first blood analysis, or you need even to go for more sophisticated techniques, imagine a colonoscopy or a resonance, whatever. What happens is that all of these techniques are centralized. Even a very basic blood test, you need to go to a centralized laboratory. And sometimes you are waiting several hours or even several days for the results. Imagine the same happening for a colonoscopy, for a resonance. I mean, we have a very excellent techniques in the clinical diagnostic, excellent, so don't worry about that. But the only problem is that they are time consuming, they need a lot of sample. Imagine, for example, in the blood analysis, they are taking milliliters of your blood. No? And you need trained personnel because finally you need technicians, you need this laboratory installation, and so on. So this is the main problem of the diagnosis that we are serving, and now that we are waiting for the diagnosis a long time. Sometimes it's depending on the disease, could be very late when you get the answer for the diagnosis and then what is the right treatment. For example, what happening now in the sepsis or the infection, that they are taking like even several days to identify which is the bacteria that is attacking the person, and then you need a lot of time to give the right treatment to the patient. And also, as we saw in the pandemic, this is really not, it's not really able to do for a very population. What you need to make millions of people screening, it's not possible to do with this technology. Okay, so what we see for the future, the solution that we propose, and is almost arriving now, is to use what we call a point-of-care biosensor technology. So the idea is the following. Imagine that you take just a drop of your blood, or a drop of urine, a drop of saliva, just a drop. And then you can deploy in a very small device, what we call a point-of-care device, and then you have an instant diagnostic. Imagine that the medical doctor, even if you go to see your family doctor and you do the analysis even just in front of him in the desk, or you can do it at home. So this is many possibilities to use all these point-of-care testing devices. Probably you have seen some people wearing this wearable biosensor. This is one of the wearable biosensor for diabetics people for monitoring glucose. Probably you have seen these people with this white cycle in the swimming pool or in the beach. And this is just one that was released by the Abbott company a few years ago, and this is measuring the glucose concentration of the diabetic people in a continuous way. So they are using just a reader, they close the reader, and they get an instant reading of the concentration of the glucose. And of course, it's very easy just now to have all this technology connected by wireless to a central station, even to your medical doctor, to anything. So probably in the future, you're going to deploy a drop of your blood, your urine, whatever, you get an answer, and probably even you are going to receive a WhatsApp and saying, okay, you have to go running to the hospital, or you can relax, this is not so dangerous. Okay, so this is what we're working in my group. We are working for since many years, trying to convince the companies, the diagnostic companies, that is the future. They never saw that like that, but now after the pandemic, everybody now is concentrated in using this kind of technologies, because it was not only during the pandemic that we need to make analysis of millions of people at the same time. It's also that during the lockdown, many people, they were not able to go to visit the doctor, they were not going to hospital, and many people they didn't get the diagnostic on time. And many people now, they have a cancer, for example, in a very late stage, or even any neurodegenerative disease, anything. So this is the main reason why now is so important that we really focus our effort in trying to do all this point-of-care diagnostic technology. But it's not only that you can do a very easy diagnostic and very fast. It's also that have to be enough reliable. So you have to be very comparable at the same level that the techniques, that the sophisticated techniques that we are using in the clinical laboratories. Okay, so this is what we call a point-of-care biosensor. So the idea is that we can fabricate a biosensor for each specific disease in such a way that you have prepared your chip, your sensor, your biosensor, and then depending on the application, you can apply the sample, these drops of urine or blood or whatever, you introduce in a very small machine in a few seconds or a few minutes, depending on the application, then you can get the result on your smartphone. Okay, so this is the idea. Many people, not only my group, but there is many groups around the world that were trying to develop, not only to develop, but also to transfer and to commercialize. We would say that this is not so easy now to understand with the industry, but to commercialize all this amazing technology, in my opinion. And I think it's going to be the real future for the diagnostic in medicine. Okay. So, but what is a biosensor? Because I know all of you know the glucose biosensor, the one that is used by millions of diabetic people in the world, but probably no, the people doesn't understand, doesn't know what is the definition of this biosensor. So in a biosensor, what you have is a sensor, a transducer, a physical transducer in contact with a biological receptor layer. This biological receptor layer is a biomolecule that is completely selective to the molecule, to the sample, to the molecule that you want to analyze in the human sample. In the case of the glucose, for example, what we have in the biosensor is an enzyme, a protein that is interacting only with glucose. I don't know if you have seen, for example, why the glucose biosensor is so specific, because in blood we have hundreds of different substances. But why this sensor is only detecting glucose? Just because the sensor has inside this biological receptor that is completely selective, only to glucose. Doesn't matter if you have many other things in your sample, in your blood sample. Okay, so this is the basic idea of biosensor. So joining two different worlds, you have something biological molecule, and you have to join with inorganic, with a physics sensor. So this is joining really a multidisciplinary approach. Okay, so the most famous biosensor is the glucose one. It was the first idea was in the year 1962 from a medical doctor in the US, and they stand for, they spent more than 30 years to go to the market. It was a very difficult device, but now millions of people are using this device every day in the world. There is also probably more simple biosensor like the pregnancy test, that probably I don't have to explain, so all of you know this. And with the pregnancy test, they are looking for a specific hormone in the urine of the women, and they have also a receptor, a biological receptor, able to recognize only this hormone. It's the same that the rapid test that inundated the market this year, the antigen test, the serological test. It's also a kind of biosensor that they are also under development or also using in the clinical practice, but there are not so many. So the idea of making this point-of-care biosensor is not so simple, because it's not only to make the sensor itself, the sensor and the biological receptor, but also, you have to deploy a liquid sample, even if it's a drop, but you have to think, okay, how I introduce this sample, so we need to do all this microfluidics cartridge. In our case, that we use optical sensor, we need to introduce light sources, for example, laser, you need photodetector, you need some electronics, and everything has to be in a very small space, because it's something similar to the pointer that I'm using now. So you have to think how to miniaturize and how to put everything together. And also, if you want to use in any place and any time, then you need also to to introduce a battery charge, and also you have to need also some wireless connectivity. So it's what we call this lab-on-a-chip integration. Okay, so in my group, if you look in there are thousands of different development of biosensors around the world. There are many people using mainly this electrochemical sensing, these electrodes, because the glucose biosensor is using that. People are using nanoparticles. There is a very fantastic new technology using just paper. where you just introduce some nanoparticles inside the paper. And this is a very low cost solution, and there are some companies now trying to push this technology, especially for using in Africa with a very low cost test, and there are many others. In our case, in my group, we decided many years ago to use the light and to use the properties of the light. And why to use the light is because with light you have a very ultra sensitivity. So we can use the light in order to see, even to monitor even the presence of only one virus or one bacteria, and that's the reason why we decided to use light. Imagine, for example, the light that is traveling an optical fiber. Everybody knows that the light is traveling in the optical fiber, in the interior of the optical fiber. So we're using this principle, how the light is traveling, there is always some part of the light that is going out in order to make our sensors, in order to make our chips. I'm not going to explain all the physics and engineering, don't worry, but just to see, I just wanted to put here some of the devices that have been developed during many years in my lab. For example, the Pointofcare Biosensor that we introduced. We make use mainly nanotechnology to do all these sensors. We play with gold, for example, and we make some kind of nano rings and nano dots. I mean, many fantasies with gold, but mainly because the light is traveling in this gold, and we can use to trap all the biological molecules. And also we use some chips, silicon chips in that case, we were mentioning that. So we're fabricating the same silicon chip that we use in the computer, but in our case, we fabricate, and then we have some photons traveling in these microchips, and then we can use also for biosensing. And you can see here that we not only design or fabricate, but you have also to integrate incomplete machine, incomplete platform. So this is making a long process to do everything. Okay, so this is something that we have been doing in my group. It's not, it's not very usual, because especially in Spain, that you develop your own technology and you make all the complete design, fabrication, assembly, in clean room facilities, and then you miniaturize and you make the hardware, the software, and all the engineering of the devices. Okay, but for me the most important is the application, because okay, the technology is technology, you can believe me, it's an amazing technology, unique technology, but of course you have to demonstrate with real application. And one of my main motivation is to show how our technology is really useful, and also how we can transfer this to real application and at the end to go for commercialization. So just to show some examples from my lab, for example, we have been working on how to make a Pointofcare Biosensor where you can use for the early detection of colorectal cancer in order to avoid colonoscopy. So this is a kind of cancer, the colorectal cancer. As you know, the only technique to detect, to make a precise detection is colonoscopy. But we know also that this cancer is one of the few that is giving an immunological reaction in our body. So when the first cancer cells start to grow, then there is an immunological reaction. They segregate some antigens, and then in response to that, our body starts to fabricate some autoantibody to annihilate these antigens. So because these autoantibodies, these proteins, are flowing in our blood, if we detect that these proteins are there, we know that a person is starting a colorectal cancer process. And according to the clinician, this process even starts 3 or 4 years before the cancer can be detected by a colonoscope in the hospital. So I mean that we are able to develop a biosensor and then make it this very easy test, because you need only some drops of the blood of the patient, and then if you can see here some of our biosensor, we just defining 3 of these antigens, and then we are just detecting the autoantibodies, the proteins that the people is developing, so we can say that this person is starting a colorectal cancer process. So this have been working with this idea, how to develop this point of care. We have already demonstrated that it's possible to detect in the serum or in the plasma of the patient with a very good sensitivity, selectivity. We have been already using clinical samples from people that are suffering already with colorectal cancer. We think that in the future it's still not finalizing, but we think it will be a very easy test for detecting who is starting the colorectal cancer, and then we could avoid the colonoscopy. That is, of course, not very nice for the patient, and also it's very costly for our health system. Another example that we have been working and is ready to be commercialized is a Pointofcare Biosensor where you can detect when if a celiac person had been eating gluten. You know that the problem with the celiac disease is that these people is completely intolerant to gluten, so the only therapy is not eating gluten at all, so there is no solution for this disease. But the problem is how to control who is eating gluten or not, because normally the celiac people say, okay, we never eat gluten, which is not true. And sometimes, there is some cross-contamination. You go to a restaurant and they say, well, there is no gluten here, but you go home and then you don't feel well. And probably there was some gluten, and we identify also the problem with the children. People, you send your children to the school, but then, you can't try to tell him or her, don't eat anything there, but of course it's very difficult to control, and probably they're eating many things, many cookies or whatever with their friends. And then they arrive home and they don't feel well, so how to make this test? So we know that when you are eating gluten, there is a very small molecule, a peptide, very, very small one that appears in urine. So when you are eating gluten, due to the digestion process, this peptide appears in our urine. So we developed a biosensor that's using a specific protein developed by a company where they can detect this peptide. The presence of this peptide in urine. And we developed a biosensor where we can just take a drops of the urine. It's very similar to the pregnancy test. But in that case, we are detecting this very, very small molecule that is also in a very low concentration. And we can detect directly in the urine. We don't have to clean, to process anything. Take the drops of the urine, put in a biosensor, and then we can see who has been eating gluten or not. And even we do some experiment with a baby from one of my colleagues. Okay, just bring the urine to my lab, and then the baby was under a diet of gluten-free. And then we didn't see anything, the presence of the peptide, but we gave a cookie and we wait 2 hours, and then the peptide appeared in the analysis. So this is a demonstration how this can be used. We are collaborating with a company, and our intention is to go to the market with this point-of-care device. Another example is, for example, for the anticoagulant syndrome. This is a commercial name that many people have to have. They have to use this anticoagulant during their whole life. And the main problem with this anticoagulant is they have a very narrow therapeutic range. So depending on the concentration, you have a very low dosage, you have a high risk of coagulation, and you have a high dosage, you have a risk of bleeding. And it's very difficult to use the correct dose for each person, because it depends on each person. So normally these people doesn't feel well, they have to go running to the hospital many times, to the urgency. So what we develop is a biosensor where we can monitor just in the plasma of the patient, and we use just only 100 microliter of the sample of the patient. We can monitor what is the concentration each moment. So this is something that they can do the analysis at home, and they can use even themselves the concentration of the therapeutic concentration. We develop also complete point of care, also in an European project, for example, for tuberculosis detection, where we can detect the presence as well of a very small molecule, a liposaccharide that is coming from the cell of the tuberculosis bacteria. And then it's only present in people with active tuberculosis, not with latent tuberculosis, active tuberculosis. And it's have a presence in urine. So it's a very easy test, because again, you can take some drops of urine, you can put on the sensor, and you can see who is infected with tuberculosis or not. And we have already demonstrated. I'd like also that you can see here. You can see here the chip that we're using. Look here. And this is a chip where we have 6 biosensors in parallel inside this chip. It's a microelectronic chip that has been fabricated here in Belgium, by the way, by IMEC. So now with using this biosensor, we have been able to analyze the sample from 20 patients coming from Tanzania, and then you can see that the urine are completely different. The color, the density, all the patents are different. But we just deploy on the sensor. We don't clean, don't filter anything. Just deploy on this very small chip, and then we can detect who is healthy or who has already suffering from, infected with tuberculosis. The analysis is less than 10 minutes directly in the urine of the patient. So it was a very fantastic European project, a very good result, but nobody wanted to commercialize. So this is a pity that finally many things that we develop in the lab then finally stay in the lab, and then it's not going for commercialization. Okay, just to finalize another example, sepsis, for example, that you know that is a very dangerous infection. We developed also some small machine. We went to the hospital, in that case in Barcelona, in Valdebron, and we tried, not only moving to the lab, to the hospital, and do the measurement directly in the hospital with our colleagues there, and then also in that case we were able to know the presence of a bacteria, E. coli, and to know who is infected, who is developing sepsis. And in that case we used only 10 microliters of the plasma of the patient. It's amazing because it's even less than what, it's a very small dose. And then we can detect, in that case we took more time, but in any case we can also try to improve this. But in any case, 40 minutes is nothing in comparison with sepsis. Sometimes they need several days to identify which is the bacteria causing the sepsis, and even the antibiotic resistant profile of that bacteria to give the right treatment. Okay, well, we have developed, my group has been working on this for many years, so as I has been one of the pioneers, not only in Spain but also in Europe, or even at worldwide level. So we have developed many different applications along not only the technology, so we developed the technology on one side, so all our technology are proprietary of our group, but at the same time we demonstrate with application, because we think always the added value is to the real demonstration that your technology is working, and it's working in the real life. And just to finalize, of course, we have been working in the diagnosis of COVID-19. My group was granted with one of the first European projects in March 2020, and we started to work with the diagnosis of COVID. As I know, I don't have to explain this now, because everybody, I think everybody knows that the famous SARS-CoV-2 and the detection of the SARS-CoV-2 is mainly done by PCR during the detection of the virus genomic and RNA. And there is also, this is the main test. There is also the rapid test, the antigen detection test, where you detect some of mainly the spike protein in the envelope of the virus. And then there is an indirect way to see who has been infected using the serological test, the detection of the antibodies that we produce as a consequence of the infection. But PCR, as I said at the beginning, is an excellent technique. But as also I said at the beginning, time-consuming, you need lab installation, they need personnel, and have some issues with the reproducibility. All these rapid tests that probably all of you have done, probably, antigen or antibody tests or serological tests, they are very rapid and they are very good, but the main problem is the sensitivity. If you have a very low level of virus, the antigen test is not able to see that. And the same happens with the serological test. That they can give you an answer that says yes or not. It's like the pregnancy test. You say yes or not, but then you need to go to confirmation to a laboratory, to a final clinical test. So what we wanted to develop is a point of care where you can do the same to see if you have or not the disease, the infection, but at the same time to make a quantitative detection. Give a number, because a number is very important for the clinical management of the patient. If you say, okay, this is the viral load, it's low or it's very high, but it's different. You can see how is the development of the disease. So we have been working on this COMBAT project, and now we finalized a biosensor for the virus detection. So our biosensor is saying yes or no, but it's also giving the viral load. And we can detect in the patients from 100, the presence of only 100 virus, that is very low, it's extremely low, to 10 to 7 virus per milliliter in 15 minutes, and it's 100% selective to COVID-19, different to flu or to common cold. We have also developed a biosensor for serological detection that we have already done all the clinical validation in Spain. And also, as I said, you can say yes or not, you have the antibodies or not, but also you can see here it's quantitative, so we can get the number of immunoglobulin that each patient has produced, and it's very important, for example, for the vaccination campaign. If if a person has a very strong immunoresponse or not, and you can see here the control, the people. We took some control from the biobank from people from the blood from 2017 to be sure that they never were in contact with the coronavirus. And you see that they give no signal, but for example, for the different patients that have been already suffering from coronavirus, you see the different immunoresponse. So there are people having a very strong immunoresponse and others having a very low immunoresponse. Okay, so we're still working on the project for the European Union. because we think that because we plan also to use the device to monitor the presence of coronavirus in reservoirs animals, especially bats. I mean, to use the machine, and this small device that you can go to the caves, to any place, to do the analysis on site, and you don't need to take the sample and to go to do the PCR as people have been doing until now. And also we think will be a very interesting biosensor to have, for example, in the emergency units, when someone arrives and they don't know if a person has COVID or has flu or has a common cold. So we can differentiate also with our biosensor very easily who is the infection that is attacking this person. Okay, just to finalize, my vision is that diagnostic in the medicine of the future will be using this kind of very small point-of-care biosensor devices that you can use anywhere, in any time, at home, in the ambulances, in the emergency units, even in the school, in the office. So probably we'll have many of these. We are going to become a hypochondriac society, to get up in the morning and to say, oh, I am going to test today for the cancer, for this, probably. But okay, but I'm completely convinced that this is the future. Yeah, to finalize, I want to thank all my people because last year we were not in lockdown, so all my people were the only group working in my lab, in my institution. So we did a lot of work on this COVID-19 detection. So thank you for your attention.
I have made many notes. It was very interesting to listen to you both. And I'm sure the audience is also with in mind plenty of questions. But I'd like to start with one, and then we can give the voice to them. How close do you think is this to us? Do you think for Liesbet, do you think one day in the hospital we'll have like a lab, or maybe we have today with technicians that will just print whatever the doctor says that person needs in the moment?
It depends on the type of implant that you want, but for instance, the dead implants that are personalized, those indeed that becomes much more standardized, and there are experiments where people try to understand they can have a 3D printer somewhere close to the operating theater and have them come out. The living implants, there is a lot of quality checks that are required. Mm-hmm. But if we can go to sensorized robotic rooms, then you know beforehand by simply reading out during the process all of the elements, all of the quality checks, then you know that what comes out is viable. And for a lot of cases where there is, it's probably not going to be very likely that there are going to be big factories where they're going to be producing these living implants en masse. So it might be that it's going to be smaller units closer to the hospital where these things are being built. Also because the costs for the living implants are quite extensive still. So this might not be the go-to solution for everyone, but for very specific cases.
Yeah, what about the sensors, Laura? Are they today, some doctors have already some sensors available in the clinic to test rapidly some things?
Yes, I mean, there are now many of these, not many, there's few point-of-care biosensors that people, the medical doctors are using, but there are few of them because, as I said at the beginning, besides the glucose biosensor that everybody can appreciate that has been something, so amazing and so useful for the diabetic people. For the rest, the clinical diagnostic is a very well-established business. So all the clinical laboratories are very well-established. It's a problem for us to try to compete with these people. But now after the pandemic, and they realize that they really need this portable technology. They need these portable devices, not only because even in the emergency unit should have been very useful to monitor constantly what the people with COVID-19 was developing. So I think now the mentality is changing a lot. Probably now the diagnostic company are looking having this focus on how to use this portable or decentralized technology as much as possible.
I have here in my mind something you said, that the glucose sensor took 30 years to come to the market. Mm-hmm. Is it now getting better? Is that this process is getting? Is it the fault of the regulation of the approval, or more the manufacturing part, or the acceptability of of the society also.
Well, in that time was because it was a pioneer idea, and then you have to join this biological world with something with an inorganic or a physics sensor, and how to join these two different worlds was very difficult in that time. The technology was not mature enough, and for example, you need all these protein, these glucose oxidase enzymes, and that time because they are produced by our body. How do you get all these enzymes and then to produce so many of these biosensors was also a problem. How to develop that? So it was the technology was not mature enough. It took a long time to be able to develop this brilliant idea in a real product. That was the main reason. And second, the regulation. In that time, there was no regulation on that. It was something completely new. So that's the reason why it takes so long time, and also because they have this biological molecule, and the biological molecule they have to maintain the activity to be alive. And the problem they didn't know how to stabilize to be alive for a long time. But now, because we learned all the lessons before, I think now it's much more easy to develop the biosensor because we have now all the expertise or the knowledge from the beginning.
I had also another question, because the sensor is something that you just use once and then you have the answer, but an implant is something that you might have for your whole life, right? So how stable they are, all the materials, and do you have also sensors to follow up on the life of the patient?
I think when we are at the point where we're going to implant, at this point, with the solutions that we have at this point, they're not being followed up with sensors. They are rather being followed up by imaging, and we're evaluating that way. When we're looking into animals, I think at this point in regenerative medicine, it's more about trying to identify how long it needs to stay there, and then find the materials that meet that demand, let's say. Rather than trying to monitor, we try to make sure that what we put in has a specific behavior. So during the animal experiment, we might actually try to use biosensors and try to use modeling and imaging to try and avoid to use too many animals, let's say. But then once with the patient, we of course, we should have shown that these materials, that we know very well how they behave, and then maybe the need for these biosensors is not there.
Okay, so you have talked about hardware mostly, but I guess you also need to couple with software, right? And so if you have a sensor with applications that will tell you the result, maybe one day on our cell phone we will have, and you have talked about many different applications. How can we merge different sensors together? Like one day we will have a computer with different applications to test different things.
Well, the idea with biosensor I didn't show here, but what we try to do is that you have a multiplex capability. So with the same sample, with the same drop of blood or urine, you can measure imaging 3 or 4 biomarkers at the same time. So this is the main idea. And all the technology we try to go at least for 2, 3, 4. Because for any disease, I mean, it's very rare that for glucose, okay, you have only to monitor one, but normally for any other disease, normally you need to monitor 3 or 4 things at the same time. So we need to make all these multiplexing capabilities. And of course, now there is also a tendency how to use artificial intelligence, machine learning. So we trying also now to train our sensors or our biosensors. So there is also a new tendency on that.
I think it's very complementary or very important as an input to the development of the digital twin of patients. Now, these were all digital twins of parts of the production process that I showed, but there is a lot of work being done also by regulators on approving this in prediction of degenerative diseases, for instance. And if even for fracture healing, if you would have a biosensors that could help you measure the level of inflammation that is going on systemically in your body, this might be an early predictor of when a fracture healing case is going wrong, and then you could intervene very early. So you can take this information, put it into a software platform. It can be something that is artificial intelligence. It can also be really a model of the fracture healing or bone regeneration process that requires certain input. And that is something that we call a digital twin, because it really talks to the physical object, the patient, through the use of these sensors.
I could keep asking questions, but maybe I will ask if the audience has some question. Okay.
So I want to ask.
Speak up, or maybe we have a microphone.
I can speak up.
Okay.
I want to ask why there is a resistance from companies towards these sensors, because to me it seems like the best business in the world. I mean, the people that have made this rapid antigen test, they're now rich because everyone has to use it. And diseases are just not gonna, I'm not saying COVID, but people get ill all the time. So there is a business there. Why they don't want or have not wanted to invest until now?
Well, but in the case of the rapid tests, I mean, these are more like very basic biosensors. There are many of them for many diseases, but as I say, for example, like the pregnancy test, but it's only yes or no. And normally, after you have done this test, you need to go to a confirmation in a centralized laboratory. It's not only that you have the pregnancy test, finally you have to go to the central laboratory. With the rapid test, for the antigen rapid test from SARS-CoV-2, in theory, you should go also to do a PCR because, I mean, depending on because they are not sensitive enough. You have yes or no, but they are not completely reliable. So the idea is, okay, this is affected because, okay, you have the rapid test, but you need confirmation. You need the centralized technique. In the case of the biosensor, for example, that has been showing or I developed, this is much more sophisticated. I mean, they are completely with a very high selectivity. They are very reliable, so you don't need to go for a testing in a clinical laboratory or a clinical or just this specialized technique. So this is the main difference, and that's the reason why. Because I mean, you have your business. With imaging, for example, for the blood, just the very blood testing. With the blood testing, I mean, there are hundreds of different laboratories around in all our cities. So this is a very good business. I mean, you are trying to replace with a very simple biosensor, but you deploy just a drop of blood and you have all the analysis. So that's the main reason why they were not so in favor of all this technology. Even if they have the sample of the glucose biosensor is saving life. Because people, a diabetic person, they cannot go every day to do a testing in a centralized laboratory.
Sometimes you really need something that is big and disruptive, like a pandemic, to put all the noses in the right direction. The mRNA vaccine is another example of that. It has been in development for I don't know how long,
20
but
years.
It took. There were within the companies, within the regulators, there was hesitancy to replace something that worked well already. So. Once you get then this big demand, then sometimes it can go really fast.
Talking about COVID, Laura mentioned that she was developing an application for detection. Do you also, Liesbet, in your lab, has the pandemic somehow changed some of your
not the research lines, the way we work, of course, but not the research lines. But I know that people involved in, for instance, there have been people working on stem cells and how to use that to develop treatments for the lung fibrosis that might occur. If we are, some of the predictions say that people in 10, 15 years time might start requiring a lot of lung transplantations. So there is also a lot of work being done on organoid systems where you take small clusters of cells to mimic organs on chip and to try and understand what is going on and find the right diseases. So people have been using regenerative medicine strategies or in silico medicine strategies to look into the pandemic, but not in our lab in particular.
Does somebody else has?
There is no question.
No questions in the chat? Okay. I had, you finished, Laura, with something that is, I see that might be a downside, like everybody with all the sensors trying to detect every morning what we have. Do you also see kind of a downside on your applications, or like more on the how it will disrupt the society or the way we see it now? Because I really see that some people will get addicted to sensors.
I think the cost for the regenerative medicine, especially the cell-based implant, is going to be very prohibitive in using this. But something similar as for the in silico component, you could have something similar that you're eternally trying to see, if I do this, then what will happen to me? Or if I do this, and this is a bit of a dystopian view on how these technologies can be used. But I guess at some point there will be some balance. I mean, if you become a hypochondriac, your doctor will put a stop to that. They will just block you or mute your signal. Yeah.
One question?
Yes, sure.
Well, thank you for your talks. I really enjoy it. And has anybody think about using the toilet as a laboratory? I mean, instead of using this small lab-on-chip, is there any possibility to do that? I mean, because you have plenty of space and plenty of samples. Yeah.
No, otro que I can incorporate in my business plan. Starting a new company. No, I don't know if some Japanese groups they have also thinking. I mean, in placing in the toilet or doing something there. I probably, because they have the more advanced toilets in the world. So I think so. I have seen something, and probably from some Japanese group. But it's a good idea as well.
And a second question is regarding the lab-on-chip you have right now. What is the limit of the number of parameters you can include? Because if you go you get a blood test, you have like 3 pages of.
Okay.
So, but in your case, how many parameters can you measure?
Yeah. So what we can measure at this moment is between 4 or 5, is not more. We call this biomarker, no parameter, because we have to think that a biosensor is measuring a chemical molecule or a biomolecule, but you have to think that you need a bioreactor that is catching the one that you want to analyze. For example, we are not, biosensing is not a technology, for example, for measuring potassium or calcium ions. It's not for this for this kind of parameters. So what we say is we are looking for some biomarkers that are indicative of this assay. So we need to, for developing a biosensor, you need to know and you have to know the clinical input. So it's not that I have to look for something that the clinical doctor already say, okay, you have to look for A, B, C, and then I develop a biosensor for that, okay? It's different. Just be careful because, there is this famous story about this company called Theranos. They have now in the trial in the US, you remember, because, this was a girl, well, this is American Dream, yes, it's called that, American Dream. It was a girl several years ago. When she was 19, she went to Stanford University. She started to study chemical engineering, but in the first year, she said, okay, I have a brilliant idea, I don't need to study anymore at the university, and I will start a company called Theranos, and she promised that she has a diagnostic technology in such a way that she was able to detect 100 parameters in 1 drop of blood. Okay, that was the idea. Nobody saw the technology, nobody saw the patent, but nobody saw nothing. But because this is America, US, sorry, not America, US, so they started the company and they got investment of billions of dollars. She was nominated in the Forbes list as one of the most rich women in the world. The company was called, and then they start to deploy this point of care technology in the world, in some of the pharmacies there in the US. But they never say anything. They just pick up the sample, take to the company, and give the result. And then some people just start to, some employees start to say that everything was fake. And they just started to send some information to the Wall Street Journal, and they were first in publishing that. And then everything was fake. They have no idea, no technology, nothing behind.
It's excellent marketing.
Excellent marketing. But
she's standing trial right now.
She's standing in trial. The trial is started in September. They expect to finish in December.
Yeah.
So there is even a famous documentary in HBO. Out of Blood in Silicon Valley, because it was also a company in the Silicon Valley, so I recommended you to see that. So that you need to show that you have a technology, a very reliable technology, and even then, even if you have a reliable technology, it's really difficult to go to the market.
Sorry, I just wanted to ask a question based on that example you said. Do you think this has put more pressure into companies to prove that you are not that kind of company, that you are working with, I don't know how to say it, real technology?
Yes, well, I think it happened. Well, for example, for me, when this woman started this company, remember I was giving some presentation in some conferences, and someone asked me, "But why are you doing that?" Because there is a company in the US that they are able to make the measurement in one drop very easily, 100 parameter, and I always say I don't believe that. I mean, I never have seen any result. I have seen no technology. I don't believe that because I have been working on this. I think it happens in the US. I don't think this could happen here in Europe. I mean, because of course, I mean, the investor at the beginning, okay, you can invest some money, but probably a few millions. But in the next round, you are going to ask, I want to see the result. I want to see the technology. I want to see something. So this is something that that is. I mean, what happened in this story is something that nobody can believe. How is possible that someone believe, especially a woman of 19 years old without no background. I mean, I think it's very important the formation. I mean, the education to go to the university, a PhD, and this is very important. I don't believe in this miracle, So,
but having cases like that sometimes put a lot of pressure on the whole development, or stop the whole development of a field. On the one hand, because people think it's already done. On the other hand, when it's shown to be fake, then the level of trust is going down so much that you have to fight against that, and that is why it's so important to do the regulatory. So very clearly, within Silico we had the exact same thing. We don't have a frame, we don't have a way of validating this that is written in rules. So we went to the policymakers here in the European Parliament saying, look, we need rules on this. And in some cases, we even got the very jaded answer of saying, well, just take it to the market, and if there's a scandal, then we'll make sure that there are rules. But if that would have happened, then we can kiss the technology goodbye for 10 years. So that's why we want to do it differently and properly.
Yeah, and that's why it's very important to communicate, right? And on the other side, it's very important that we scientists learn how to communicate well and how the people can trust what we are selling.
There's a question
there.
Yeah, talking about communication and transfer of information. Elena in the chat is asking that to both of you, how do you see the knowledge transfer from science academia to the companies or clinics in Belgium and in Spain? And if there are difficulties into knowledge transfer that should be addressed with new policies.
I can maybe start here, and I'll talk from the perspective of the in silico medicine, where we have international organizations that happen to be both based in Belgium. One that is an academic society where, in contrast to other scientific societies, we from the beginning have been working on this transfer from the computer screen to the patient, knowing full well that there are hurdles that we need to take as scientists, we need to do part of that work ourselves. And there's a second society where we have teamed up with industry, because it is really important for some organizations like the EMA that you go there as a scientist, but for other organizations like the Parliament or the Commission, it's important that you show that this is a link with industry. And then together we can work on policies, because policies of course are more important for industry than for academics, but it's really good to have these people there to make sure that there is this whole continuum of people and of profiles and of interests that are sitting around the table working on establishing some things, because it's only when all the people are aligned and when everything is there, the policies and the regulation and the industry and academia, that you can really have a good transition of bench to bedside.
Well, according to my experience, I know that here in Belgium, because I've been involved in many evaluation panels, even for Flanders area and so on, I know that the connection between academia and industry is much better here in Belgium than in Spain. In Spain, there is no connection at all. But there is one. I will be more polite, I will be more politically correct. There is some connection, but it's not working so well. So we really need to do much more effort to make this connection between the academy and industry. And it also have to be done by our politicians. And in Spain, this is not very good, even if there are many different ways where you can apply for this collaboration between academy and industry and so on, but it's not very well organized. And this is a problem in Spain. And we saw also, I was even complaining to the government, because during the pandemic we saw that in Spain we didn't produce any technology at all. I mean, we didn't produce any antigen tests, any serological tests, any PCR. We know how to do the PCR, but even all the reagents and even all the machines you have to buy outside of Spain. So we saw during the pandemic there was nothing. I mean, the technology, we know how to use the technology, but we don't develop the technology, and this is a real problem as a country. So this now there is a reflection and trying now to make some restructuration and try to get a better connection between academy and industry, but still we are far away. I think from Belgium or from other countries like Germany or England, for example, doing that. And also there is many academic people, this is also something for the scientists, because many scientists, and this is also especially also in Spain, they don't want, I mean, they think, okay, I do just blue sky or doing science, I don't want to poisoning my science just with industry, and this is also a mentality that we have to change.
And I think maybe there's another group of stakeholders that is becoming more and more important, that is the patients and the citizens and also the clinicians, of course, because now when you go to a regulator with an ATMP or with a new therapy, the regulator will ask, did you include a patient organization? And if you ask for funding, did you include a patient organization? The story of Rose is one that came from a Bone for Kids, which is a patient organization that tries to collect money to fund translational research for these large bone defects. So making that effort to communicate to them what we are doing with taxpayers' money, what we want to do to help them, that is also becoming increasingly important, and not just as a token or as a lip service, but really it is adding. It can help push things forward if you have the patients behind you.
Yeah, well, it's a final question perhaps. Yes, you have mentioned this critical problem of communicating, have evoked it in various ways, and you have talked of the science engineering end and industrial end, the regulatory end. But I am missing something which is patient in a way is example of what I mean is citizenship. So communication towards this background of policy making, which is the public opinion. So what is importance of tackling this very challenging mission of informing so that it is not only concerned patients but society in general that helps these. You mentioned a number of things, very regretfully so, remain forgotten in the lab. So sometimes it's because there is no social pressure to ask or demand for that, or perhaps, of course, the regulations prevent a very simple or very cost-effective solution to that. But wouldn't it be, or isn't it so that one of these challenges is precisely informing the citizenship in general?
But I think that one of the main problems, in my opinion, is that they pretend that the scientists, we are like an orchestra woman. I mean, that you do everything. And if I do science, I do technology, and I have to also to educate my people in the lab and everything. I cannot do everything. And the problem is that they pretend that we do everything, that we start companies, that we do science dissemination, that we take account, speak with the policymaker, that we know all the regulations. So sometimes it's too much. I mean, you cannot do everything, or not everything properly. So I think that we need to have a much better assembly where, for example, if I have very good products in my lab from my research, I need a team, a specialized team that they are able to take that and then to transfer to a company, to speak with company, and so on, and not myself. And that's the main problem, especially in Spain, that I have to do everything. And this is not possible. I mean, not possible at all. I mean, you do
everything is too much.
Everything is too much. And also, for example, I have a large group, more than 20 people in my group, but probably for you is the same. So, but we have to provide the contracts, the money, the labs. I mean, you have to do everything. It's like to have a small company, and then you have to be all time with your headache, how to get money enough to maintain the contracts, to maintain the people, to maintain my lab. And beside that, then you have to think in the tech transfer, and you have to think in the science dissemination. So it's too much. So I think we really need to have a more stable system.
And sometimes it's difficult to find the attractive message, because science can be really slow, sometimes excruciatingly slow, or you can go and almost reach the point where you think, now I'm ready to go to the patient, and then you get a setback and you have to start all over again. So then that means that you can't communicate every year on something that is new and exciting. Also, when you have a good study that you published and you go to the press office of the university and you ask, well, I've written this press release, can you, but it's not very spectacular. They always want you to solve cancer in the next pill that you designed, which is not the case. So there needs to, I think, also be some information. Somehow it needs to be made clear also to the public, and it's not through their fault that they don't realize that always, that science has its own pace, and sometimes it is slow, and sometimes it looks like for 10 years we haven't produced anything, but sometimes maybe it makes a small contribution to something else where you don't expect it, and that also is an important contribution. So communicating is fine as long as it doesn't need to be flashy and 140 signs every other week.
There was a quick question maybe?
Yes. You are producing different in vitro devices, and you are talking about the commercialization also. And I wanted to know also which is the best strategy. So the group is talking with the companies, or the best is to try to create a spin-off with the support of the university. And if this is support of the university, or if this support is.
Well, my strategy is to start my own company. So I started the first one in 2004. So I must say I was one of the first in starting spin-off companies in Spain. And I sold the company a few years later. So it was a very successful story because it was bought by Mondragon Corporation in the Basque Country. So my young idea is trying to generate a spin-off company to grow the company, and then to see if you can join with other big industry. So this is my main strategy. And now we're starting a new one this year as well. So we have some private investors and so on. But again, a lot of work is done by myself. This is what I'm claiming. Even if you have tech transfer unit in your institution and so on, and everybody is willing to help you, but finally you have to do a lot of this work. And what I think is that it shouldn't be done by the scientists. Of course, I am never in charge of the company, so I just started the company, but I have a specialized CEO person in the company. It's separated from my research group. This is my strategy. I did already in 2004, a long time ago, and this is the best strategy. This at least is the one that I'm following, but I would like to have much more help. I mean, help in the sense that if I have very good technologies, I think it has to be the tech transfer units or my institution, whatever, that they commercialize that, and not myself.
Okay, so with this I think we can conclude the event. I have few points here to finalize. So I guess the future of medicine and technology in medicine will be to deliver a personalized medicine, centralized in the patient, right? Accessible to everyone, low cost. And we cannot forget the trust in that, in the technology, the regulations, because they are very important, so we don't repeat the story Laura just mentioned. And of course, better structure to communicate and to have more help, a more solid structure to transfer the technology we do in the lab to the companies and to the society. So with this, I would like to thank Instituto Cervantes, Consejería de la Embajada de España, and our sponsor also Fundación Ramón Areces, all of you here present, all the people online, and of course our stars tonight, Liesbet and Laura.